Okay, so today we will do the topic variation from Mendel's principles. You know that Mendel has given certain principles up to which you have understood and learned, and today we will learn about the variations of the Mendel's principles. And in this variation of the Mendel's principle, we basically to understand that, we have to understand certain terms. Some of the terms which you already understand is that locus in a chromosome. Suppose a homologous chromosome is there, then there are one particular locus where a particular trait or the gene for a particular trait is located, from where protein or set of proteins, maybe one gene or a set of genes are located here, or from where the protein or a set of protein will be expressed through central dogma of molecular biology, and due to that expression there will be showing of certain trait of a certain characteristics or character into the organism, or as the phenotype of the organism. Up to this much you have learned. If you have understood my previous videos, you will understand what I am saying right now. And if there is a tall plant and its homologous chromosomes looks like this, then this locus codes for the coding for the tall phenotype of the height character, and this will code for the dwarf phenotype of the height character. But however, the combination of capital T and small t will show a phenotype of tallness. Now the basic thing is that these two are the alleles, okay, alleles of a one particular gene, the gene which is responsible for a character, and the character which have two traits or more than two traits. Examples also you will get now in the upcoming topics. But yeah, so this concept is there. Now the fact is this: based on this locus, when this locus interacts with this locus, or this gene interacts with this gene, or I can say this allele interacting with this allele, we call it IntraAllelic Interactions, or we can call it IntraGenic Interaction. And there can be another example or another thing that you should understand. Suppose, let us take example of two homologous chromosomes, and suppose here the allele of a particular character is there, and here the allele of another character is there. Okay, when these alleles will interact with these alleles, okay, when the interaction between these two is known as intraallelic interaction or intragenic interaction. But when these two will interact with this, these two will interact with this, or these two will interact with this, we call it interallelic interaction or intergenic interactions. So please keep in mind, this understanding forms the base of understanding incomplete dominance, a variation from Mendel's principles, CoDominance, another variation from Mendel's interactions, multiple allelism, polygenic inheritance, and epistasis. So in our course of videos, we will try to understand these 5 topics, which are actually the variation from the Mendel's principle. I hope that you understand what I have said up to this much. Now let's little bit move forward and try to understand what is incomplete dominance. Now when we try to understand incomplete dominance, for understanding incomplete dominance, we have to Let us take the example. There, look, three examples that we should have to keep in our mind and remember. One is of Andalusian fowl. Another is of the Mirabilis jalpa, which is also, this is the scientific name, which is also known as 4 o'clock plant. And another is Antirrhinium, which is also known as the snapdragon. Okay, so these are the three examples that we will understand about incomplete dominance, or keep in mind about incomplete dominance. But that doesn't mean that incomplete dominance is only shown by these three. But these three are there in our syllabus and we will understand that. Now, what is incomplete dominance? Let us take example of either Mirabilis jalpa or Antirrhinium. Almost same pattern of incomplete dominance is shown. Incomplete dominance is shown by them. Now you see, in Mirabilis jalpa, 4 o'clock plant or Antirrhinium, there can be three types of flowers available. We can see three types of flowers in these two plants. Number one, we can see a variety of white, which is the recessive variety. We can see a variety of red, which is the dominant variety. And we can see a variety of pink, which is actually the incomplete dominant variety. Now, let us understand this, how and why this is a variation from Mendels' principles and how we can interpret that. Let us try to understand this. Let us take, according to Mendel, let the P1 generation be taken as a dominant red flower showing plant and is crossed with a recessive white flower showing plant. So this is a red flower showing plant. This will show red flower, and this will show white flower. So when the red flower and the white flower will be shown, the red flower is crossed with the white flower, which is the parental generation. Now when the parental generation, male and female or female or male are crossed, then you will, if I try to find out the F1 generation, and if I do it through a Punnett square, then I will get this. Everybody knows that. But however, according to Mendel, you will expect these plants to show red flower, right? But no, all of them are going to show pink flower. Now what actually is happening is that in these offsprings of the or in the offsprings of the parental generation or in the F1 generation, when we see the homologous chromosomes containing R and R, you, according to Mendel, will expect that R or the proteins produced from R will mask the small r. Either it will remain unexpressed. That only you have to understand, that according to Mendel, this should remain unexpressed, and this should code for proteins or pigmentation which will give red color to the flower. That is according to the Mendel. but in originality that is not shown. In originality, pink flowers are shown. When in originality, pink flowers are shown, you will have to understand that, okay, that means intra-allelic interaction that is happening is not in accordance to the Mendel. Is not in accordance to Mendel, because here some protein products must be produced from this and some protein products must be produced from this, and both of them must be produce a mixture of pigment which is giving a pink coloration. So both are expressing in such a way, both are being expressive in such a way, but both the pigments are expressed. Now, in such a case, the R cannot completely dominate over small r. The capital R cannot completely dominate over small r. So, it is called incomplete dominance. Now, when this is called the incomplete dominance, I hope that you understand that you get an intermediate character in case of incomplete dominance. You get what? You get an intermediate character. You neither get the dominant character nor get the recessive character, but get the intermediate character. In case of incomplete dominance, you get an intermediate character. In case of incomplete dominance, you get a intermediate character. Not the dominant, not the recessive, but the intermediate. Keep this point in mind. You get the, where should I write it down? Here I am writing down intermediate character. Okay, so you get the or intermediate phenotype. Now, if I try to find out the F2 generation, if I try to find out the F2 generation, how it should look like. The F2 generation. Now, in case of the F2 generation, RR, that is a pink colored flowered plant, should be crossed with RR, a pink colored flower plant, and RR will be the gametes and RR will be the, and you will get RR, RR, RR and RR. Now here in the F2 generation, you obtain a red flowered colored plant, white colored flowered plant, and you obtain pink colored flowered plant. Now, when you obtain such cases, in this you get, if I try to find out the phenotypic ratio of F2 generation, you will get 1:2:1, the phenotypic ratio. Because why? One flower will be red, is to pink, is to white. So red is to pink is to white, you will obtain 1:2:1, which is the phenotypic ratio. Now, if you go for the genotypic ratio, in case of the genotypic ratio also, you will obtain 1:2:1 as same, because one will be RR, two will be R and another will be RR. Now, if you can remember the simple crosses previously have learned, the F2 generation phenotypic ratio was 3:1. Genotypic ratio was 1:2:1. Now just notice that the phenotypic ratio has changed, but the genotypic ratio remains same, which is quite justifiable. That okay, the inheritance of the genes in the chromosomes is same. It is the same pattern. Then sir, why are you calling it a variation from the Mendel's laws or Mendel's inheritance? I am calling it an inheritance from the Mendel's laws or Mendel's inheritance because of the reason that I can see that in phenotypic, first reason is that I cannot see in a heterozygous condition dominance of the dominant gene. However, I can see incomplete dominance, first thing. Second thing is that in the phenotype, in the genotypic ratio, I can see 1:2:1, which is same, but in phenotypic ratio also I can see 1:2:1 because 2 I will obtain pink colored flowers. Okay, so this is the difference, where in normal Mendelian inheritance you will find 3:1 of the dominant and recessive, but here dominant, recessive and codominant 3 only you will find. I hope that you have understood. Many a times in your competitive examinations or your clever teachers may ask you question that in a type of cross we find same genotypic and phenotypic ratio. In a type of cross we find same genotypic and phenotypic ratio and blah 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 blah, then blah 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 what? Question will be asked, that is true, but when this hint will be given, we find the same genotypic and phenotypic ratio. So same genotypic and phenotypic ratio is found in the F2 generation of the cross of a type of organism which shows incomplete dominance. Remember that. With this hint, you will understand that incomplete dominance is being shown. You have to understand that incomplete dominance is being shown. This particular thing you have to understand. Now that is example of Mirabilis jalpa and Antirrhenium. Let us do for Andalusian fowl. Just hold on for a second. I'll right be there. I am just correcting my condition of my pen. Just hold on, boys. Okay, so I'm back here. So let us understand about the Andalusian fowl. Let us understand about the Andalusian fowl. Just give me one more minute, okay? I'll just adjust the light. Okay, thank you for your patience. Okay. Now talking about the Andalusian fowl. Now Andalusian fowl, there are black varieties, there are blue varieties, and there are white varieties. Now let us take that the black varieties to be the dominant ones. Let us take the black varieties to be the dominant ones. However, does that matter? Let's not just let's open up our minds little bit. Okay, let us take the black variety as capital B, and the white varieties as small b, small b. Now, when you cross this, you will find that there will be BB in the F1 generation. This obviously you can understand will show blue. And when the blue varieties are crossed among themselves, you will get black, white and blue chickens or fowls. Now, which is understandable that okay, fine, it is fairly understandable, it is just like that only. But you know, I would like to tell a story here which is very, very interesting. You see, some people who were associated to poultry farming, okay, once this happened, that people who were associated with poultry farming, they thought that the black, this happened to be a trend, okay, and the white ones, the black and the white ones were sold cheap, but the blue ones were considered delicacy. Now, what the poultry, they used to send the blue varieties at high price, and people used to buy also. Okay, so this happened to happen. Okay, and then what happened is, this poultry people, what they did, they thought that why don't we keep the blue ones only and exhaust the black and white ones, fatafat say, and don't keep them, because they give us less money, blue gives us more money, and this happened to be the thing. So what they did, they exhausted the black and white ones, fatafat. Okay, and there was no black and white, they killed some, okay, and there was no black and white available in the farms. Now what happened, when they crossed the blue ones with the blue ones, which were actually showing incomplete dominance, they only got 50%, 1:2:1. So if 1:2:1 is the phenotype, 50% of the chickens or the 50% of the fowls only showed blue colored, and 50% they got back blue and black and white. Obviously, that was a blunder for them. Okay, and then obviously they were not students of biology and couldn't understand that if they would instead had kept the black and white, they would have always obtained blue variety. So just a story that I shared. So I hope that the concept of incomplete dominance is understood by you. Next, I should move into codominance. Incomplete dominance is done. Next, I should move into codominance. Okay, let's see. Codominance is little bit different from incomplete dominance. In incomplete dominance, we see a intermediate thing, right? Intermediate phenotype. But in codominance, that is not the case. One good example of codominance is coat color of cow. We can see that the skin of the cow or the skin of the dog or some dogs shows a kind of pattern that in the entire white skin there are black patches, or entire black skin there are white patches. Do you know about Dalmatian dogs? Okay, there also you can see this, where two colors are expressed simultaneously. Means, suppose for example, if there is a black cow crossed with a white cow, now you get this cow will not show a grey color, which is the incomplete dominance. It will show maybe what a cow. Okay, it will show maybe white color, means both are interacting. So that means if I consider it to be the two homologous chromosomes, and B stays here. In case of codominance, proteins from here are also expressed, proteins from here are also expressed, and both are expressed individually. It expresses its phenotype, it also expresses its phenotype. Nothing can be denied, or a mixture of the phenotypes cannot be expressed. Remember, in case of incomplete dominance, no specific gene is there. Okay, suppose if BB is showing blue color of the fowl, neither B is showing blue pigmentation, neither small b is showing blue pigmentation. It may be so from these alleles, two proteins are synthesized, or rate of synthesis of the protein may be more or less due to the presence of each other. The intraallelic interaction will be somewhat like that. Because suppose for example, think that if this is shown for the fowl, neither this iska protein nor iska protein will show blue, black and white दिखाएगा. लेकिन दोनों अगर इंटरेक्ट करके दोनों मिल के कुछ मेटाबॉलिक पाथवे से, या दोनों ऐसे इंटरेक्ट किया कि दोनों एक साथ एक इंटरमीडिएट कैरेक्टर दिखाया, तो इसका एक इंट्रा एलिलिक इंटरेक्शन है. और जब कोडोमिनेंस होता है तब इसका एक इंट्रा एलिलिक इंटरेक्शन है, जिसमें क्या होता है कि दोनों का प्रोटीन सिंथेसाइज होगा और दोनों फेनोटाइप दिखाने के लिए राजी होगा. तो इसका कोई स्पेसिफिक जीन है ही नहीं, नो स्पेसिफिक जीन. बट कोडोमिनेंस में there exists the presence of this, which are both specific genes for codominance. So I hope that you have understood the difference of incomplete dominance and codominance, and both are variation from Mendel. So yeah, that is it. Another example of incomplete dominance is that blood grouping. Now, not all the blood grouping, because all the blood grouping is comes under multiple allelism, which we'll learn next. Okay, but in case of blood grouping, you see when there is a person whose blood group is AB blood group. Now, in case of AB blood group, if a person is showing AB blood group, his RBC in his gene, the locus will show IA and IB. Both are expressing the proteins, and both will be expressed. So the RBC will have both the antigens on its top, the A antigen, the B antigen, both embedded to its cell membrane, cytoplasmic membrane or cell membrane, embedded both the proteins, because both are showing. That is codominance. This is codominance. The AB blood group is also an example of codominance. So I hope that understanding codominance was not very difficult for you, and you have well understood the things. Okay, so with that I end this video. Go into my next video to understand about multiple dominance.